altogether there are 11 genes that we have to put into a rice plant, and it has taken 7 years to clone and transform each one of those, transform it, make sure it's stable, go through maybe 5-6 generations, so 3 or 4 years, produce a plant that expresses those. Then we take 2 plants that express the gene, we cross them with traditional crossing so that both genes are in one plant. And we have done that again and again and again. This plant here now contains 11 genes, so all of these that you can see in purple and in green here. So to best of my knowledge,
what generation this is?
This is an M3, so the 3rd generation, that contains all 11 genes, which is, to the best of our knowledge, the first time that 11 genes have been crossed into one plant. But much more than that, those genes actually function, and they're in the right location. So here we can show that not only have we transformed them, but we have to get them in the right place, functioning at the right time, in the right way. And so you can show here, this is an insert into the leaf, and you can see the veins here and the cells across the veins, and this dark staining shows the expression of those genes within the leaf. And you can see that they're just in these cells around the edge, not here and not here, and in this one a different gene just along these cells and not here. So those 11 genes are functioning and they're in the correct location as well. That's another picture of that. Now, with this 11 genes, it is a partial C4 plant, but if you compare it to this plant here, which is a wild type plant, no transformation, you can see it looks very similar. And the actual truth is that with 11 C4 genes expressed in the rice plant, they really look very similar. They grow in the same way, they behave, and they produce the same amount of yield. And this is actually a positive, and it's a positive because although we've made it partially C4, it is not complete, because it doesn't have those anatomical changes.
What generation this?
This is also an M3.
No, it's the front one.
Now this one I brought in to show you. This is a single transgenic, so it's one of the transporters, and you can see that it is very sick. This is also a 3rd generation plant. So this gene is already in this plant. When you express it on its own, very sick. It survives and grows, but quite sick, pale leaves, very low photosynthesis. But when you put it together with all the other genes, the phenotype disappears, the plant is healthy, and it kind of recovers. And so what we've been now trying to do is work out whether we've been successful. Are these genes, is the C4 pathway actually functioning? And that's what you can see in this apparatus here. This is an apparatus designed here to measure the activity of that C4 cycle that I showed you. And what we're able to do is clamp a leaf in here with a cylinder behind and remove the CO2 from the atmosphere, and then inject our own CO2. And what we do, if I go back to our diagram, is we've been trying to assess whether these genes are functioning. We know they're in the right location. We're trying to work out whether they are functioning. So what we do is supply labeled CO2. We let photosynthesis happen, we put them under light, we pass the air through. Then we bring these 2 bars down, we freeze the leaf, we grind it up, we run it through a mass spec, and we measure the CO2 in each of these enzymes. And if we do that, what we can see is that there is a greater incorporation of CO2 in these C4 enzymes, which suggests that this plant has a partial photosynthetic function, a partial C4 function. It's on its way to being like this plant, but very small. About 5% of the photosynthesis in this plant comes from C4. But this is very encouraging because it's the first time that we've done it. So how now do we move from this plant to here? Well, we think it's the anatomy, and what we now have to do is reproduce. Let me show you. What we now have to do is change the leaf from this to this, and we think if we do that, then this plant will begin to behave more like this, and we will get the higher yield. Now this, I said, was very, very difficult, changing this, and what we started with at the C4 project was actually by creating populations of these. So we would take sorghum, we would irradiate it with radiation, or we would put a chemical on the seed. We would grow those out in the field. Here we started with 1.6 million plants of sorghum that we had irradiated with radiation, and then we grew those out. We took leaf samples like this, and then a lot of people sat around looking through small microscopes, and what they were doing was counting the veins on the leaf here, and they were looking for alterations in the leaf structure that would look like a which would look like a C4. So here is a C4. You can see the big veins here, and you can see them in green here. In this mutant, you can see this is a sorghum plant, and you can see two things. When you look at the leaf section here, a greater distance between these veins, and you can see it also here. So by looking at the leaf surface, we were able to identify mutants with changes in this anatomy. And it was hoped that if we could do this enough times, if we could look at those 1.6 million plants, we would identify plants like this that had a loss and a change in this anatomy. Then what we would do is we would identify the gene, and we did that by sequencing. So we would take the DNA from the plant, send it for sequencing, and then we were able to identify within these mutants what had changed in the genetic code and identify what controlled that anatomy. With those genes in mind, what we've ended, or what we're doing now, is cloning those genes and then transforming them into this rice plant. And the hope is that if we do that, then we will get this anatomy, something like this, along with the C4 function in here. Now, I've said a very positive story, and it is, but it is not that easy. This has proven to be very, very difficult. In this screen, from the 1.6 million plants that we screened from sorghum, and we did another 50,000 in rice, we found two genes. And it turned out that they were both the same gene, identical in those two mutants. But it was not a gene that led to a stable change in the anatomy. It was a secondary thing. So what happened is we mutated a gene, it affected something, the anatomy changed. When we put it in rice, no anatomy. But we didn't give up, and we haven't given up, and I think you know this. We also tried in rice, so in this field next to us here, we did the same in rice. This time we were looking for a C4 anatomy, but this time nothing. We found nothing. And so the strategy now has changed slightly, and I think you're already aware of this. So now the groups in Oxford and in Cambridge, they're taking a slightly different approach to see if they can identify this anatomy. Instead of taking a plant, trying to break it and make it work, what they have been doing is they've been taking these plants, they've been cutting them up, and then they've been looking at both the expression of genes and saying, where are these genes expressed? And what they do is they look at mature leaves, they look at very young leaves, and they look at how genes change as the plant grows and the leaf grows, and they try and work out which genes are switched on when this leaf grows. The other thing that they have been doing is looking at plants that are neither rice, like rice or sorghum. So you'll be familiar with C3, C4 intermediates. These are kind of evolutionary stopgaps, if you will. They were originally like rice. They for some reason started down this process of being C4, but then they stopped, and they're not a fully C4. So now people look at those and say, can we compare the genes of these, this, and something in between, and work out what controls the anatomy? This has been going on now for the last 3 years, and the paper from Jane Langau has reported some success in that, so there is a hope that we will identify some more genes. But we still have a lot of work to do before we can put that into this rice plant. There is also, and I guess this is quite important as well, a number of other things that we need to be able to do. some things that we don't know about and some things that we do. So if we look at the anatomy, other things that would need to happen, in a what rice, these cells here that are very important in a C4, they're bigger. In rice they're very small, so we need to make them bigger. We also need to make them functional. In rice they're empty or almost empty. They have no function. So somehow we need to put all of the machinery that is needed for photosynthesis into these cells. But we don't know how to do that. One of the other things that we would need to do here is that the cell wall that surrounds these cells, in C4 very thick, stops gas leaking out. But in rice very thin, so we have to make those thinner. Here, these cells here, they are between the veins. You can see they're like little fluffy clouds, they're highly lobed. This is a function of photosynthesis in rice, but is not good for a C4. So we have to make these cells round and non-lobed. And I can honestly, hand on heart, say, at the moment, we do not know how to do this. There may be other things that we do not know how to do, and here's the tricky part. We don't know what they are, because no one has ever tried to do this. So we have to discover and find. We are 7 years into what we said would be 25-year project. So we have a good progress. We have made a start. We have a plant that is looking like a C4. We begin to figure out how to do the anatomy, but still much work still to do. And then obviously if we were successful, and your colleague was asking what would happen, how would we create this plant and turn it into a product that we could release outside. This one we could not. This is a research plant. It's transgenic and many alterations probably. So once we figured out how to do this and we can get rice to do this, then we need to think, how do we make this into a plant that people can grow, farmers can grow, that we can release across the world, across Asia, because this is an IR64 that grows very well here in the Philippines, but not so well in Bangladesh, certainly not in India, not in other parts of Japan. So once we know how to do this, we would need to do this C4 engineering in different varieties. But that would come at the end of this research kind of phase. I'm sorry if that was a bit too... Thank you very much.